After about a week in the greenhouse, the buds start to open. As soon as the leaves are the size of a small coin, they can provide a sufficient amount of DNA for many molecular tests. The DNA is extracted by undergraduate student researchers at the biology department at Missouri State University in Springfield.
Right now we're collecting samples for testing with DNA markers. The technique we use is polymerase chain reaction, which amplifies from a very few nanograms of DNA. So from just a few nanograms, we can make many, many tests. For one, it's extremely accurate, and also you can send it from scientist to scientist without worrying about the restrictions. For example, DNA doesn't carry diseases, whereas if you were sending plant samples back and forth, there would be a lot more restrictions.
For DNA extraction, scientists use a streamlined procedure that enables them to quickly and easily get enough DNA for all kinds of molecular analyses. The DNA will be tested with molecular markers, a standard and highly versatile procedure. This technology can be used to follow the inheritance of agronomically important genes, determine the relatedness of different grapevines, and to study the genetic basis of disease resistance in North American native grapevines.
So we put our samples in liquid nitrogen to freeze them, and in a minute we're going to pulverize them to break the cells open and get at the DNA that's inside the cells. And we only need a little bit of DNA that this method gives us so that we can run a polymerase chain reaction on it and generate more of the molecule that we need. So after we pulverize the DNA, we're going to add some chemicals to prevent the oxidation of the DNA. And then after we add those chemicals, we're going to heat it, which will fully disintegrate the cell, but the DNA will remain intact because it's highly stable. And the cell remains can be centrifuged away from the DNA, and we'll collect it.
Molecular techniques enable grape geneticists to rapidly identify disease and pest-resistant genes in wild grapes in order to introduce them into cultivated varieties. The resulting new generation grapevines will require a considerably lower input of pesticides. Their cultivation will be less harmful to the environment, and their products will contain very low levels of pesticide residues, if any. The biological basis of this new viticulture is the vast genetic diversity of North American wild grapes, which developed resistance to the devastating pests and pathogens over evolutionary time as a means of survival. Hermann Jaeger and his American and European colleagues recognized this in the 19th century. Today's grape breeders and geneticists rely on the same immense biodiversity of our native grapes, the result of millions of years of evolutionary product development. Just as the wild grapes in the Missouri Ozarks were essential for Hermann Jaeger to develop his own grape varieties, they are important genetic resources in the development of a new generation of grape varieties in the 21st century.